So welcome everybody to the next, I guess, Memory Decoding Journal Club. If you've been here before, you know what we're about. If you haven't, you'll find out in a moment because we're going to be discussing the criteria for the Memory Decoding Prize, at least somewhat. And today we're discussing Sammons et al. It's a 2024 Proceedings of the National Academy of Sciences paper called Structure and Function of the Hippocampal CA3 Module. This is not a new paper to this journal club. In fact, Ken Hayworth previously presented this paper and its findings in one of our early 2025 journal clubs. When I was thinking about what to present this time, seeing as last time I was presenting something very broad, an overview of the hippocampal system and what was known about it, what was potentially the consensus, I was actually considering a set of different papers I could present today. And there were at least 4 good standout papers in there that could have been today's discussion. Some good contenders, but I decided to revisit this one now with the subtitle, How Does a CA3 Connectome Study Win the Memory Decoding Grand Prize? And let's hope this moves nicely. Okay. And here's why. And I'm just going to bring back this slide one more time as a refresher. So the Aspirational Neurosciences Memory Decoding Challenge is, if you just read out the line that's on the front page of the website, the first demonstration of the decoding of a non-trivial memory or other learned function based on only a static map of synaptic connectivity. And with some input that we received or that we kind of digested from last year, this time, in the early part of this year, we aim to propose a clear set of example studies that could win that grand prize, or a description of a proposal, sort of proposed studies, how they would be set up, what they would show, and why they would win the prize then. Now, one animal model and brain region that's a prime contender is the decoding of autoassociative and sequence memory engrams in the connectome of the recurrent fiber network of region CA3 of mouse hippocampus. Here, this is from last time, so our last memory decoding journal club. This is figures taken straight out of the Lau and Lasonsi 2024 paper. These are representations of the anatomical and functional mouse hippocampus. Functionally, sensory information is processed by neocortical regions. Then via entorhinal medial temporal lobe, spatiotemporal neural activity containing regular patterns of spiking elicit activity in hippocampal areas. And hippocampal region CA3 is right at the receiving front end of these pathways and crucial to episodic sequence learning. You can kind of see it's right here, receiving input. You can see that the complete hippocampal network, if you look at that, is fairly complex with multiple input sources and internal recurrent pathways. Prior functional experimental work, including in vivo work, slice work, demonstrated a notable prevalence of recurrent activation via connections from CA3 excitatory pyramidal neurons onto other CA3 excitatory pyramidal neurons. And that led to common models of hippocampal function, including models like the Rolls and Treves models that you were just referring to, Ken, in which recurrent fibers between these excitatory cells in CA3 carry out essential autoassociative mapping of meaningful items, or engrams if you want, during learning, and mapping between these engrams to form sequences. Now, in some models, CA3 is more involved with autoassociative patterns and not so much with the sequence recall. In others, sequence recall is the main task of CA3. So there's a little bit of back and forth in these models between what the assumptions are about what every region or the connections between regions are doing. But the involvement of recurrent fibers as a very important part of memory encoding and recall is clear throughout all these models. And pattern completion during recall happens through those recurrent excitatory connections between CA3 pyramidal neurons. So why this paper? The Sammons et al. Publication here is a great example of the type of work that's necessary for a successful memory decoding study. It's not what we would call a winning study, but it moves along in the right direction. The parts in the study are clearly headed in that right direction, and so we could use that, say, as a concrete case study to ask, why does this particular study not win the prize outright? What else do you need for a clear win? And there are some pretty obvious points. In part, the aim of the study is quite different. So at what point could you say, here's a demonstration of decoding a non-trivial memory based on a static map of synaptic connectivity? Now, the combination of steps in the Sammons et al. Paper covers three important bases. Collecting a, by today's standards, large enough sample of data that produces a static map of connectivity, so a connectome in the relevant area. And for this, they are utilizing a multi-beam scanning 3D EM, and they acquire high-resolution 4x4 nanometers in the XY and 40 nanometers in the Z image data. That's within the plane of CA3 pyramidal neurons in mouse hippocampus. They process that data, identifying cells and reconstructing dendritic and axonal arbors to identify pyramidal to pyramidal excitatory synaptic connections. And that's how they then obtain a mouse CA3 to CA3 connectome sufficient for their aims in their study. They also carried out extensive patch clamp experiments, or extensive, I guess it depends on comparing with what paper, but extensive enough patch clamp experiments in CA3 using slices from 52 mice. And that provides functional verification of some of the assumptions and results that went into the EM portion of their analysis. The patch clamp experiments were used to confirm levels of connectivity, which was important for several reasons, among them to ensure that results obtained from EM were not an effect of biased dendrite selection, and to ensure that the results were not badly skewed by failing to distinguish between active synapses and silent synapses in EM. They also backfilled neurons with biositin as yet another way to trace connectivity and then measure distances. It's another secondary confirmation of the connectivity statistics they found. And finally, they conducted a computational model or modeling study to evaluate the effects that different levels of CA3 to CA3 excitatory connectivity would have on the size of memory engrams that are needed to achieve successful auto-associative recall and sequence recall, and therefore also the memory capacity of the 1 million or so neurons, pyramidal neurons in mouse CA3, under the assumption of sparse hippocampal memory encoding. So these three experimental bases, EM patch clamp and computational modeling, are what makes this a great case study. But you have to take note that the goal of the Sammons et al. Paper was to confirm or dispel hypotheses about the degree of recurrent connectivity that exists in mouse CA3 and consequent effects on the size of individual memory engrams and memory capacity. This paper did not explicitly aim to identify specific memory engrams to extract or decode memory. So we have to consider what steps would be needed to take or need to be taken to do that then. Also, we need to consider how the EM functional and computational studies would need to be modified or extended in order to satisfy other requirements of the memory decoding challenge, such as, for example, that the decoding can be said to rely on the static map, but that what was decoded constitutes actual and potentially even meaningful memory engrams in whatever way that can be established. So they're engrams that really are engrams, not just potential engrams composed of silent synapses that could be recruited at some final or later date, but ones that are actually involved in some memory. And that the result is non-trivial, in the sense that the autoassociative memories or sequence memories can be differentiated or distinguished with a number of decoded items that demonstrates a reliably detailed method. So let's begin by reminding ourselves of the content of this paper, with its mission to provide reliable results about the degree of CA3 to CA3 connectivity in mouse hippocampus. And I'll do that by, I guess, talking about that a little bit first, and then we'll get into the discussion. Sammons et al., in their paper, they were responding to a set of earlier anatomical results and functional results published in Science by Guzman et al. In 2016. In fact, much of what Sammons et al. Did was purposely replicating the Guzman et al. Study. Guzman et al. Also carried out multi-cell patch clamp recording from up to 8 pyramidal neurons at a time in vitro. They also carried out biosite and labeling and tracing, and they carried out simulation studies using fully reconstructed pyramidal neuron models. What Guzman et al. Could not do back then is they didn't have the ability to do a large volume reconstruction from EM. So the connectomic study by Sammons et al. Is able to give direct and explicit insight into the connectivity network that was not available to Guzman et al. The Guzman et al. Study was surprising at the time because it found a probability of only about 1% that any two of the pyramidal cells they were able to study had excitatory connections between them. Such an unexpectedly low connectivity led them to consider higher order motifs where interconnectivity depends on intermediaries. And this is something they showed in their modeling study. Now, while they showed that that could still achieve associative recall, that dependency runs counter to sparse coding models for the hippocampus and didn't align with the previous consensus of connectivity assumptions, which turned that science paper into a potential disruptor of the theory. Using EM reconstructions, Sammons et al., the paper we're looking at now, found cell-to-cell connectivity probabilities an order of magnitude greater than Guzman et al., which brings it back to, in fact, what they found was between 8% and 10% probability, which is in line with typical models of sparse engram encoding in CA3. Okay, so I've got a link in here that I'm not actually going to visit just to save on time, but this would be back to the same WebGnosis link that Ken also used when he presented this paper. If anybody wants to, explore the data themselves, we can provide that, of course. I'm just going to go through the figures. So here in A, you can see that they were working with a volume almost a millimeter in X and Y with a depth in Z of 62 micrometers. That's kind of a, I guess, rectangular pancakey slice. It's not as large as the cubic millimeter data sets that we're now commonly seeing coming out of the Allen Institute in Princeton, but it's quite significant. The volume it's a good volume. The volume size is, though, not sufficient to contain the full arborization of the pyramidal neurons that have somas inside the volume. This means that some of the potential connectivity can't be determined directly by tracing when the dendritic or axonal fibers cross the volume boundary. So the connectivity that Sammons et al. Established in this paper should potentially be considered a lower bound because of that. Now, their tracing and connectivity calculations, the results of which are mainly shown here in D, are restricted in the following way. They found 7 pyramidal neurons for which the traceable axons had at least 2 collaterals. That means those axons had at least 2 branches off of the main axon root so that the axons can make connections with neighboring neurons. They identified all of the outgoing synapses on branches of the 7 axons, and that amounted to 1,062 synapses. From those synapses, they followed the postsynaptic dendrites in search of target cell bodies, or the borders of the data set when that happened. They determined that if a target was another excitatory, they determined whether a target cell was an excitatory cell, mainly by looking at the spininess of the dendrites as a characteristic indicator, since pyramidal neurons generally have more spiny dendrites than, for example, the interneurons you would typically find in that region. Of the cell-to-cell connections, 96 out of 947 were multisynaptic. I'm not sure if that made a huge difference here, but it's worth noting. They didn't observe any autopsies, so connections from cells onto themselves, and it's not clear exactly why that is. And then they limited their analysis to the excitatory connections, of which there were 795 with target cell bodies within the volume. They also measured the cell body to cell body distances so that they could construct statistics comparing connectivity with distance. And that's what you see in the graph in D. The gray lines there are the number of connections at a particular distance for each of the 7 source cells, and the black line is the average of those. And the result that they found was a connectivity of about 11.2% within 50 micrometers, and that dropped to about 5.5% at 250 micrometer distance. The actual connectivity can be lowered by the inability to determine in EM if a synapse is silent, but the actual connectivity could be greater because the calculations are based only on the connections that could be traced within the volume. So it's probably still a lower bound, despite that other caveat. Then the patch clamp study used. Sorry, is there someone asking something?
Yeah. Can I jump in on
totally
the
jump in?
slide?
Please do.
Sorry. So I had it was on the previous slide.
Okay.
So I have a couple of questions. One is how to think about them choosing cells that have axon collaterals in their volume. And I know my initial thought of this is that could cause a big bias because it's like they've chosen the ones to give the most connections. At the same time, if they're thinking about what is the number of connections in 3D, then choosing something that would have a slice of that 3D that actually has aligned with axons from these cells is probably a good thing for them to do. Could you just address what impact choosing these cells
before I do that, can you repeat the first part where you were a little bit worried that picking them was a bias that's problematic?
So immediately they just say, okay, so we performed this study based upon these 7 is a low number, right? These 7 neurons who had 2 axon collaterals that stayed within our volume.
Yep.
I think I've gotten that right. Tell me if I don't.
That is what they say. Yeah. Yep.
Okay. So the immediate thought is, oh, sample bias. That's a sampling bias. And I'm wondering if, because I can convince myself that this is not really affecting their results. Even if you were to forget about the stuff that you're about to talk about with the electrophysiology that really does provide a good solid control. Just looking at this EM connectivity, should we be worried about that choice?
Yeah, so honestly, I didn't think about it as much as you did, because I honestly thought this was just a feasibility bias, as in there is a bias in there, and we don't know, or at least I hadn't really thought through whether this would be a problematic bias. But it felt to me like the choices they were making were based on what they felt was feasible during their study. So first of all, choosing 7 source pyramidals to trace, rather than just saying, okay, we're going to look at all the pyramidal cells we can find in here, and let's just trace them all out and see what all the connectivity is that we can find. They could have done that.
But clearly, if they had chosen 7 at random, they would have gotten much lower numbers.
Yeah, because those would have had axons that just depart from the volume right away and make basically no connections. Yeah. So that would have definitely pulled down the connectivity from the study. But I have a feeling that what they were doing is they're saying, okay, these axons happen to go in a direction where even though they may come back into the volume, right? So if this was like E11 and you could give them all DNA tags, you could find out if those axons come back into the same volume that they've been imaging. But because it's EM, you can't do that. And so, yeah, they're just gone. So then how do you tell if those axons actually still make connections somewhere or not? So that makes those, it's just infeasible to draw any conclusions based on those axons where you can't see the collaterals within, the volume right out in front of you. Or am I wrong?
So I think the question is the number that they get for probability of connectivity.
Yeah.
Can we consider that that this is still kind of a lower bound on that probability of connectivity?
Yeah, so I was going back and forth in my head about whether this is a lower bound or not a lower bound based on just gut feeling of, is there more of a problem that you're picking something specific or that you're ascribing real connectivity to silent synapses? Or is it more important that a big problem with the study, something that's really hard to do, is to even trace anything significant that remains within the volume? And I don't know off the top of my head if that's the main problem. I was imagining that when they don't see axons with at least two collaterals, since, I mean, what is one collateral? That could be it. Why not one collateral, actually? That's a good question. Why two? Yeah, because, I mean, they could have done that too. I wonder if there was a reason why they had to have two collaterals. But if there were no collaterals, then it wouldn't be very useful at all because it's just an axon that disappears right out.
Yeah.
I don't know. Yeah.
The other question I had is, and this is a dumb question. So if there weren't any axons coming into this, so there's a whole bunch of axons coming into this from other regions, and presumably we can't figure out where those synapses are actually coming from. And there's synapses coming from inhibitory cells within this region, stuff like that. But if we did know how to exclude those, then presumably you could do this study by just saying there's this many synapses and there's this many cells.
Yeah, so you couldn't trace them back necessarily to the cell where the axon was coming from, but you could then just say, I don't care. I'm just going to say there are this many connections between axons and dendrites. Yeah.
I guess what I'm saying is like, is there a way to do this without connectomic tracing? And to the extent that we have to do the connectomic tracing, it's because we don't know something else about how many synapses are being formed onto these cells from input axons.
But it seems important to me to get some insight into how many connections a particular pyramidal cell will form. Because if you're just counting connections, you don't know if this is some really skewed thing where you have a few neurons making an innumerable number of connections, most of them making no connections at all. You have no idea what the distribution is then. And even here, I mean, just looking at the connectivity from 7 pyramidal neurons is still darn small sample.
Yeah. Okay. In any case, those are my questions. Just
yeah, but the way I ended up thinking about it after going through the different phases of this study, when you look at the EM and then the functional and then the model, and then thinking about that in terms of what does this give a memory decoding study, is that really the EM is where you're starting to try to identify something specific about this cell, its involvement in an engram, its involvement in sequence recall. Now, this study here doesn't go that far, but that's where the EM would come in, is the identification of those things. Whereas things like the connectivity ratio are, and therefore also the pattern size that you'd expect, are something that functional studies could be better suited for as sort of the input assumptions into all of this. Or even just having done enough studies in numerous other papers beforehand, other EM studies, other functional studies, to give you some idea of what the typical connectivity is in mouse, and then apply that to a memory decoding study. Of course, this doesn't apply here because this is one of those studies trying to establish the connectivity, but that's where I ended up going in my head about, where do you get the assumptions from? How do you apply them? But we'll get into that as we go further through this. Okay, let's see, where were we? We were at patch clamp, I think. All right, so the patch clamp study. So this was used to demonstrate that the connectivity found is, or at least they tried to demonstrate that it's characteristic, so that it's not biased by the selection of dendrites, although they said nothing about selection of axons. They used a batch of 56 mice, from which they collected 119 usable slices containing CA3 pyramidal neurons, each slice being 400 micrometers thick. They recorded from 8 neurons at a time, and a total of 410 neurons. And the way they would do this is they would deliver current injections to a specified neuron and then detect consequent excitatory postsynaptic potentials in the other neurons. In that way, they tested 1,172 possible connections and found 103 effective connections, so ones that actually seem to show EPSPs. And so if you divide 103 by 1,172, that gives you a connectivity probability of 8.8%, and that was in line with the connectivity found via the EM study. Here in this B subgraph, you can see the morphological reconstructions they also carried out based on biocytin backfilling of the cells, and the biocytin injections made the axons and dendrites optically visible and traceable. So by filling the same neurons from which they recorded, they were able to obtain distance measures as well and carry out a similar connectivity by distance analysis. Not shown here, that analysis found a flatter curve than the graph that we showed for EM, though the reasons for that are a bit unclear to me, as is the impact on connectivity findings. I'm not yet sure how much we need to worry about the particular distribution in that graph. I think it matters for things like number of patterns in the entire region and their distribution and the ability to create chains and things like that. But yeah, we can get into that a bit more later. The figure also shows some examples of the current injections and consequent neural responses and target EPSPs. And C shows a statistic of EPSP numbers by their response amplitude, so how many of them were how strong. It's interesting. These are interesting details, but it's not immediately clear to me that these functional details impacted the relevant outcomes of this study. You could imagine that you might develop some assumptions based on that about connectivity strength or the likelihood of essentially silent synapses by distance, which could then be applied to the EM reconstruction. Anyway, both of their EM and functional results are a good fit to the predominant model of CA3. Now, I am going to skip figure 3 because it was essentially a replication of the study of different connectivity motifs done in Guzman et al., and it's neither highly relevant to their results because the connectivity they found is perfectly adequate for sparse coding, nor is it necessarily relevant for our discussion of a memory decoding study. But, I'm willing to be corrected on that. So then the modeling study, figure 4. Sammons et al. Built leaky integrate and fire spiking neuron models in Python to test the effects of different connectivity probabilities and to evaluate n-gram size and memory capacity given the connectivity they found. Their model was composed of n excitatory spiking neurons plus a set of n over 4 inhibitory spiking neurons. a spike timing dependent potentiation trainable connectivity from the population of inhibitory neurons onto the population of excitatory neurons was used to bring the system into balance. This means the excitatory recurrent connectivity between the pyramidal neurons was able to achieve pattern completion, but without runaway excitation, producing stable sequences of spiking output patterns. I'm not going to get into the details about how tricky that can be, but it's one of the things I was playing with a lot just recently, trying to build an auto-associative memory in our BrainGenix thing, and it took a while. The model did not include any further intricacies of the real hippocampal system, such as modes driven by rhythmic theta oscillation or the influences of different input pathways. So when you look at the kinds of results they're generating in their model here, you could consider the sequences of spike patterns produced by the model as somewhat analogous to sharp wave ripple patterns that you would observe during recall and quiet waking or slow wave sleep states when a mouse is consolidating memory, or during rapid behavioral responses. In an actual mouse, N, the number of excitatory cells in CA3, would be around 1 million. And the figure here shows tests with 50,000 and 100,000 as the N. The assumption is that a memory engram would be composed of some subset, those are the little triangles shown here, M, like M neurons, interconnected via strong recurrent excitatory connections. Due to autoassociative and sequence recall roles of CA3, if we assume that both are present in the same system, one would assume a connectivity matrix that exhibits, and now I wish I had drawn the matrix, that exhibits representations of both these strong autoassociative connection patterns as well as somewhat weaker connections between engrams A, B, C, and so forth in a recallable sequence. So if you were to sort the cells by their involvement in an engram, cluster them together, that means that the connectivity matrix would show strong connectivity squares along the main diagonal, as well as weaker connectivity squares off the diagonal, potentially weakening the further one engram is from another in learned sequence, as in temporally distant from one another. If you provide a stimulus cue that is some portion of M, then the autoassociative recurrent fibers rapidly recall the full engram of M active cells, and reactivating a few successive engrams in this manner can produce enough activation between connected engrams to recall a full sequence. They didn't really do an explicit study of, say, whether recalling a single engram ought to recall an entire sequence, or whether 2 or 3 would be needed as, say, a small cue of a sequence to recall the whole sequence. There are other papers that get more into that. But yeah, so there are nuances here. Anyway, you can potentially recall a whole sequence from a partial cue to one of those engrams, or maybe 2 of them. A log distribution was used for the background connectivity weight in a subset, and of course then these were strengthened within M and also between different engrams, so you get that representation in the matrix that I was talking about earlier. And the figure, it shows how the overlap of input activity from multiple subsets can produce the excitation needed for sequence recall. Now, Sammons et al., they would inject something like activity into half of the subset of M cells. And then in B, you can see what resulted for 2 different combinations of connectivity and pattern size M. So in one case, we have an example where the recall does not successfully recall a sequence. It sort of dissipates. And in the other one, you get a nice strong recall of a sequence of patterns, sequence of these engrams, shown here as just the neurons, and here shown as the excitation. So the difference between those 2 cases was, as I mentioned, the combination of connectivity and pattern size. We can see that also here. We've got these numbers 1 and 2 in here, which correspond to the 2 cases. So the boxes 1 and 2. The blue color hue shows the percentage of successful sequence recalls as a function of connectivity and pattern size. And you can see a clear dependence of successful n-gram size M on connectivity probability in both 50K and 100K network examples. It's not at all surprising, of course, and it simply confirms the assumptions underlying models of memory encoding in CA3. But it does allow us to make some predictions about the expected n-gram size from experimentally determined connectivity. And in the case of sparse coding, that then allows you to estimate something like the number of simultaneously encoded memories that are possible in CA3. There's a lot more that one could study, such as comparing the degrees of sparsity and overlap between memories. But, we don't have to get into all of that right now. Flattening of the curve over here in D. This shows that for connectivity 8%, which is what they used here, any pattern size M at or above about 1,200 neurons can produce reliable recall, assuming that the connectivity between n-grams is sufficient for the size of the network. And E is pretty obvious as well, in that it shows that larger synaptic weights allow for smaller pattern sizes. I mean, who would have thought? For those of us modeling similar systems at greater biological detail, there are a few things that you can do to estimate some of these variables from observable or measurable data. For instance, knowing the maximum number of ion channels at the synaptic membrane surface, or that can surface to the membrane, and the maximum current at strong LTP can give you an upper bound for single synapses by surface area of the synaptic area or spine dendritic volume. And then from that, you can derive the number of synapses you actually need to drive a successful postsynaptic spike activation of the cell body. And then all of those things you can package together and work with. If you want to just play around and find out what would work, you could, for example, experiment with our BrainGenix platform, which generates neurons through neural morphogenesis to produce a realistic systems, or at least a realistic looking morphological architecture. And then you can play around with what those systems would do, and then carry out corresponding EM calcium imaging electrophysiology. So if you want to play with something like this, there are definitely lots of systems out there, and we have one too. Okay, we've gone through the paper now. So I'm going to get into the discussion part, and please, you can already interrupt me before we even get started, or I can just finish up and then we can talk about it. I don't mind either way. This paper tells us something about how to plan a successful memory decoding study. Maybe not everything, but a bit. Let's say you conduct a functional study, either via patch clamp or calcium imaging, and from that, you can infer some probability of connectivity with one group, for instance, in the CA3 pyramidal neurons. You can measure a distribution of connectivity over distance. You can also derive the likely minimum pattern size, M. We just saw how you can do that. And using these measurements and derivations, you can determine in approximation what the EM volume size needs to be and how many neurons and synapses need to be contained in that volume to lead to the likely identification of successful n-grams, and by extension, sequences of n-grams that recover episodic memories. In addition to using connectivity information to derive size M and volume requirements, you can do some additional studies to predict how sparsity and overlap affects robust recall, and establish the actual overlap or sparsity of patterns in CA3. The more overlap there is, the larger M has to be for robust recall of specific n-gram. And similar metrics apply for the recall of sequences and their dependence on K, let's use a new letter for that, preceding recall patterns. So this overlap versus sparsity aspect produces a little bit of tension between robust autoassociative recall and sequence recall. In part, this is dependent also on the inhibitory effect of the population of interneurons and the comparative dynamics of pyramidal cell activity and interneuron cell activity. Again, things you can measure functionally beforehand. So extracting autoassociative versus heteroassociative patterns during memory decoding this is a kind of an outline of what you might have to do for that. For a clear depiction of n-grams and sequences, It's helpful if you can also sort neurons, and this is, again, fairly easy to do in functional recording where n-grams pop up. But sorting by clusters of connectivity is also possible, just in a map of synaptic connectivity. So there are ways to do that with or without function. So we have some possibly spicy requirements for connectome study, whether done by EM or optical means, but not undoable. Then there's the question of functional study requirements beyond their utility in establishing connectivity. The studies here rely on patch clamp, and it's hard to capture complete n-grams of M neurons and even harder to capture sequence activity that way using patch clamp. A calcium imaging study might be better. Studies in slice still suffer from boundary effects, though those are possibly negligible for large slices. And requiring something, okay, do we require something beyond CA3? Like something in the neighboring regions like entorhinal cortex, dentate gyrus, or even associated behavioral references. Now, I tend to worry about things like, was the modeling overly simplistic, but I think we should think more about these issues first. So how much do the extracted memories, if we're going to talk about them as memories, need to be associated with some meaning, known to mean something specific? Is it enough to show a sequence of activated n-grams with calcium imaging and then find the same thing directly from the connectome, even without knowing what each of them are for? I'm sort of tending in that direction, but I'm not sure if that is the right way to look at it. What we would like to see, or sorry, yeah, go ahead.
I'm sorry, I don't want to stop you in the
Please,
middle of stuff.
feel free to interrupt, because this is now just putting points down on paper about stuff I was wondering or worrying about and brainstorming.
Well, I'm just from what you're saying here, I just want to put something in perspective, because when I think about this memory challenge, I think of it also relative to the field. And the model that you're putting forward here, the model that they're putting forward here, I guess the model that people have been putting forward for decades, is there is no study. This is a question. There is no study that has done enough connectivity in a single a single CA3 to show that there are collections of ensembles of n-gram cells that are connected to each other, multiple of these, okay, and much less that they're connected in sequence.
Yeah,
so
that's my understanding. Yeah.
Okay, so I mean, there's a bunch of hints of that. This study is kind of the statistics are big enough so that we could maybe this could be right, right? Which is way far away from saying here is a with just larger volume.
But
if they did
this could be right only applies to the statistics, right? Not that any n-gram was seen or retrieved.
Exactly. It could be the case that they would get a connectome of CA3 that was large enough to show the thousands of neurons and their interconnectivity, and see and reorder the neurons in order to see groups of connectivity and see absolutely nothing. They could absolutely get that result, and nothing that we know really rules that out.
This gets, yeah, you're right on top of one of my main concerns when we even think about the whole way that the memory decoding challenge is specified, is that even if we say that it's non-trivial, as in there's some n number of things we want to pick up on, we could even just look at n as 1 or m as 1. What does it mean to pick up on a single n-gram? Do we need all of the neurons in this n-gram, and we need to prove that this is the full thing, it's robust? Or do we just need to pick up on some of them? Is that enough to say you've captured an n-gram? You found some neurons that seem highly connected with one another and likely belong to an n-gram? What is the cutoff here?
I mean, I'm just thinking that, you know, if there was a connectomic study, which is totally doable. I mean, if the microns had been pointed toward the hippocampus as opposed to the visual cortex, or if this study had acquired in such a way that they could do more complete connectivity, and not just what they could trace essentially manually, they could presumably have gotten a study that shows these reorder synapses give you blocks along the diagonal, like you were describing in the connectivity matrix. And if that was overlaid with just a few calcium imaged ensembles or sequences, even better, that matched. I feel, I'm putting this out there, I feel like that would be a winner.
Yeah, so a study that
and
really
it's-
reconstructs enough neurons to build up a matrix like that, and having to show that the engrams shown in this matrix along the diagonal are of a size that, given a known connectivity, would be sufficient for recall. These are all parts of it, right?
Yeah,
Yeah.
that's right.
Okay.
That's right.
Yeah.
And along with the calcium data, that would
And there, yeah, and so calcium data also, because you could be picking up on potential putative connectivity for some future recruitment of something in hippocampus that could be an engram, but that was never actually used as an engram, or hasn't been shown to be used as an engram, because they could be silent, right?
I'm not sure if I got that, but
Okay. If you're not able to tell from the EM reconstruction whether some of the synapses you're seeing are silent synapses versus active synapses, strong active synapses, if you can, fine, then no problem. But if you're not picking up on that, like this study didn't, then even if you see something that looks like a potential engram in the diagonal, may not be one that we have any provable evidence for that it's actually an engram of something, that it's pointing to something. And that actually leads to another question then, because if you can show that you found something in CA3 that could be recruited as an engram, is that enough, even if you don't know if it's ever been recruited or has been associated with some behavior or associated with something? Because you could do a behavioral study alongside it, do some of that beautiful tagging that we've seen in other papers, so that we get tagging during learning or during retrieval, and then
it's also,
see if you can find those as well.
Some of these theories would suggest, I'm thinking of, anyway, some of these theories would suggest that these ensembles and sequences are there just waiting to be used, but they're kind of natively built in anyway.
Yeah. So the question is really, is it good enough to find out that we have a RAM chip with a bunch of bits that each mean a byte? And that would be like the study, yes, we found the RAM. You can store bytes in here. Or do you need to show that there are pieces of the RAM being used for something?
And I guess, and that's probably a rhetorical question, but people can give their opinions. I'm going to give my off-the-cuff opinion. My off-the-cuff opinion is that if a paper that did all of these things ran across my my desk, I would think, wow, the models that have been proposed for decades, and that have been waiting for really strong evidence of their reality, they've finally gotten it. And so anything that wins a memory decoding challenge prize should also be a kind of, holy crap, that really moved the whole field forward in some way. And I think, in my mind, that's a better criteria than the number of bits of memory having been decoded. It's that it
but
really
even if what it's doing is it's giving anatomical confirmation of what the model is describing as how a memory can be formed and how it's used. So the confirmation that those models are resting on something really solid by giving you that connectomic reality of it. And in that case, yeah, this is so important to be able to show that that is there, that it doesn't really matter as much whether you can connect it back to these memories specifically having been active memories at any point in time.
Yeah, that's my opinion.
I just wanted to make it clear. I think your opinion is very valid. I just just wanted to be absolutely sure.
But push back.
I think just showing that there is RAM would be, I mean, in my view, insufficient to win the prize. I think we'd want it to be able to be linked back to some sort of confirmatory behavior function.
But even if you can say the RAM is organized in bytes, like there's these n-grams of size M, just like you've got 8 bits in a byte, that's insufficient for you.
I think you'd be like tantalizingly close, in my view, to it. But I think it's, at least for me, I would want it to be grounded somehow, just to sort of like assuage the idea that we're not picking up some other intrinsic organizational phenomenon or other property that, for whatever reason, doesn't end up being linked to actual behavior at the end of the day. I mean, I would be shocked if you got that close and it didn't end up being, because it would match so well with theory. But I think some sort of behavioral link would be important. But
so Ariel, I like the idea of zooming in on CA3 as an extremely concrete model and just assuming what we've got. So let's say somebody did CA3, they did thousands of neurons, they got millions of synaptic connections between these. There was a clear ensemble pattern with sequences, and let's say they had calcium data, and the calcium data was some of the ensembles were corresponding with some of the calcium firings and sequences of firings seemed to correspond with the ensembles that they were seeing from the connectivity. But let's say it wasn't. Let's say they were looking for place cells, but they couldn't. These sequences weren't place cells. They couldn't tie them back to a behavior. Would that not cross the threshold? Because now you got you know that they actually fired together as a group, and in sequence maybe, but yeah, you don't know what they mean.
Yeah, I think this would be, for me, and again, like I can strongly see the case for not doing this, but for me, if it's a memory decoding challenge, I would still want like at least even 2 behaviors at that stage to distinguish between, like whether it's 2 different place cells, 2 different locations, 2 different contexts, anything. It would be absurd. Like I could imagine some very funny version where like you have a huge connectome, you're distinguishing maybe like 100 different ensembles or n-grams or something, and I'm like, please just give me like 2 different place cells or like 2 different contexts to distinguish, and then I'll believe like the whole 100 and set that you've got elsewhere. But I worry that if we don't ground this in some way back to behavior or memory, what people think of typically as memory, rather than just the like internal dynamics of the models that we think encode memory, that would be, we could still, very unlikely, but we could still somehow have captured the wrong thing.
I am worried about that too, but I can see from the perspective of an experimenter that adds a whole extra level of complication, because otherwise you can just purely focus on CA3, just on those slices, just as those pieces, whether you're doing calcium, whether you're doing EM, whatever you're going to apply to it, you're just focused on that one little area. And you can say, yep, I've seen calcium activity. So these are they're doing something together. They're probably memories. I don't know what they refer to. I don't know what behavior or what place cell or whatever, but they're doing something. And then I also see them in EM. So there are at least 3 different stages here. Either you're totally happy with just what the M can say, and then how it blows our minds that this is confirming our models, because we can see these these RAM bytes basically in there. Or we have the stage where we say we need calcium, because then at least we can confirm these are groups that are actually being used like that, and it's not always 2 of them or 3 of them or some coordinated whatever, something else going on that our models did not predict. And then the next step would be, okay, you've got behavior linked to it as well, or some kind of sensory input. This 3rd stage difficulty level, even better grounding, getting even better that way. But then setting aside that, those 3 stages, I'm wondering if this study in CA3, should it be, can you satisfy it by finding just auto-associative patterns, just the M, just those things, or must you also find sequences? I could argue either way.
And you hinted at this before, Randall, but a lot of models, correct me if I'm wrong, a lot of models suggest that those sequences require another area.
Yeah, there's no consensus on this.
Heteroassociative.
Some models say that can all be done in CA3. Other models say, no, you need an interplay between multiple regions. So, yeah, the model consensus is not there.
This isn't answering your question, Randall, but I'm even thinking back to the previous question we were just asking. I'm actually not even sure I would require, again, if I was solely judging these things, that any detected engrams from the EM of one mouse or the calcium imaging of one mouse, that this behavior has to come from that same mouse. I think if you could show somehow that the ensembles that you recorded in CA3 were like definitively, let's say, like a place cell or place field, and like, no, like we're sure it's a place field, we're sure it's not a contextual field memory or another
let's
random
dig into
sort of
that
thing.
a little bit more, because of course lots of studies have been done where there's been a demonstration that yes, behavior and say running around in a T-maze produces place fields, and these place fields pop up and appear in CA3 and CA1 in multiple regions there, that you have some cells that respond to specific places. That's been done. So we have that confirmation. What we don't have is we don't necessarily have something where you've shown that, oh, and it's these 1,000 neurons that fire for that place field. Although, again, something fairly close may have been done using optical methods, where you can get some idea of how many cells pop up for a specific place field. So you could do that. It gets harder the more you have to make it match finding a whole M in a sense, but maybe not impossible. And maybe the standards of this study should be that high, and then being all the way through from place field to CA3, calcium imaging to EM. That could just be that strict, or say you've won this level of the prize, or you've won this other level of the prize. You don't get 100,000, you get 50,000, something like that.
Yeah, it's a good question.
We could even, when we present this as a proposed study, we could mention all 3 of these possible levels and say that, you know, this would be really cool. This would be absolutely awesome. There are different ways of writing that up, but then you're already diluting it a bit because you're not saying, here's the study that would solve it.
Well, I know that I'm thinking of one particular study at Genelia where they do really massive amounts of calcium imaging in hippocampus, I think CA3, where they are looking at how the population dynamics evolves over a learning period, and they can see that it bifurcates into representing the world on this part of the task. It's a virtual reality task, or this part of the task versus this part of the task. And so we definitely have studies that do stuff like that, and that tissue could be farmed into a connectomic study, and then you would have massive population dynamics right on top of that connectomics data. So I don't think it's that big of a an ask to actually have that type of behavioral data before account.
Okay. And would it be absolutely necessary for them to farm the same tissue where they did that optical study into the connectomic study, or could it be from two separate mice?
No, I think it would have to be. Oh, okay, so I don't see how it could be done without being from the exact same mouse, because to me, and maybe this is where I'm ignorant, to me all these cells are identical. They're all the same. Yeah.
Yeah.
Yeah. Well,
But
that
if you could do the optical study well enough that you can show, okay, it looks like M cells fire in response to this place field, then you could take that M and move it on to another mouse potentially and say, this is what we should be finding in the connectomic study. Or if we only find a subset of that, then we're only looking at this much of an engram. Potentially. But then again,
yeah,
they could have a large variety of sizes. You know, we don't know if they're very constrained to the minimum.
I share Ken's intuition that I imagine it being extremely difficult to do without using the same animal. But I think be willing to give, or like, I think something could win the prize as long as it had any degree of semantic information that was being extracted. And I'd say that like somehow if you could know that like these M cells were very much a signature of a place cell, even if we don't know like which place cell in this actual mouse out of the field of potential place cells there were. As long as you were able to say it was a place cell, and these are the sets of possible place cells, that would probably satisfy me. But yeah, it seems a lot easier to do if you're using the same mouse that you've previously done calcium imaging in, for example.
So I really think, I know we're coming to time. I really think we should take the information from this study and from Randal. You have you kind of understand the number of cells and the number of connectivity and stuff like that, and rough out that hypothetical connectivity matrix and that hypothetical calcium imaging maybe, and say, okay, this is what we're thinking of. Because I know that a lot of people that would listen to this are kind of like, okay, I don't see it. I don't know what they're talking about. This connectivity matrix with blocks, and I certainly don't know the calcium.
But just so we're clear, because I agree with you, we should like diagram it out. We are, it appears, aiming for basically the strictest interpretation of this prize, right? From what Ariel has said, from what you've said. And I'm not opposed to that. Ken, your insight at Genelia seems to say this isn't actually that hard to do. This is totally doable. So you could get this functional study, and we could put the same mouse tissue into a connectomic study, and you could get the whole thing. So then you can be that strict about it.
I think so. Although I can imagine that somebody would be patting me on the head and, oh, naive person, the calcium imaging is too blurred out in time to see on this level these engrams. I can imagine that I'm missing stuff at multiple levels.
I don't agree with that.
Okay.
Using phase precession as a way, like, because when you're doing the functional study, you might not have to restrict yourself to sharp ripples. And you could, and even in sharp ripples, we might be able to pick up on some phase precession. As long as you can pick up on phase precession, even if you have a blurring out and it's harder to distinguish the individual engrams of a sequence, you could still pick up on when they drop off and new ones come in. So you could still figure out this is a sequence and this is the order of the sequence. So I'm not sure that the calcium would be a problem.
Well, maybe we should do a paper at some point on the imaging side, since we're really locking in on a connectomic study needs an imaging side along with it.
Yeah. So do you guys have a? I guess we need to figure out two things. We need to figure out when are we going to sit down and diagram out how to do this very explicitly based on what we just discussed. And I hope we'll get a transcript of this. That's Richard. Listen, please. We need a transcript that we can diagram out what we just discussed. And then also the question is, who's doing the next paper and what's the next paper on? So 2 questions.
We can chat on Friday. or
Okay,
Thursday.
so on Thursday, we'll chat about the diagramming that thing out. And then also that's when we decide on the paper, or do you already know what you want to present, or either anybody?
I'll have a candidate by Thursday.
Okay, good. Then does anybody else want to chime in with ideas, thoughts, anything that popped into your minds as we were discussing this? What would it take to win before we close? I guess we're done. Well, in that case, I just want to thank everybody again for showing up, keeping us company, motivating us. See you all later. And
Thanks, Randall.
Richard,
It was fun to discuss.
Randall.
yeah, thanks. Richard, thank you. You can close the stream now, or close the recording.
Bye all.